Hello everyone, welcome back to Study Room. In this video, we will be talking and studying about nutrition in plants. So let us see what kind of nutrition does plants have and how is this process taking place. So nutrition is basically divided into two types, that is autotrophic and heterotrophic. So let us see what autotrophic nutrition is. Autotrophs use inorganic sources in the form of carbon dioxide and water to produce simple forms of food. So autotrophs basically make their own food, okay? They make their own food. From inorganic sources, right? So you have to remember this thing that they are capable of making their own food. So they have this machinery in themselves to make their own food. And the examples are green plants and some bacteria. So as you all know that plants make their own food and hence they are known as autotrophs. Okay. So that is why we can always give an example. Whenever you will have to distinguish between autotrophic and heterotrophic organisms, you always have to mention the definition in points and then you have to give an example so that it is very easy to understand that what it is and what all organisms actually have this kind of nutrition. Next up, we have heterotrophic nutrition. So heterotrophs do not synthesize their own food. As you know, they do not have the machinery in themselves to synthesize their own food and hence they indirectly depend on food produced by autotrophs for survival. They need complex substances for survival. So that is why they break these substances down using biocatalysts called enzymes. So you'll get a question that what enzymes are, and you'll have to define enzymes. So you'll always write that enzymes are biocatalysts which breaks complex substances into simpler ones, which help in breaking actually. They help in breaking complex substances into simpler ones. And the example that you will give for heterotrophic organisms are animals and fungi. So as you all know that we humans come under the animals category and we cannot make our own food. We derive our food from autotrophic sources like plants and hence this is a very understood thing that we the humans are heterotrophic and we cannot synthesize our own food. We depend on the food produced by autotrophs for survival. Heterotrophic nutrition differs depending on the type and availability of food material as well as how it is obtained by the organism. So this is a very important thing that heterotrophic nutrition depends on the type and availability of food material. For example, if we talk about fungi, they break down the food material outside the body and then absorb it, like cuscuta. So this is a very good example of a fungi called cuscuta. It is also known as amarvale. Okay, so the common name of cuscuta is amarvale. So what does it do is they break down the food material outside the body and then absorb it, grow, and hence cuscuta grows as parasites on other plants. Okay, so what they do is maybe this is an organism, this is a fungi. Okay, if you just take an example, this is a fungi and these are the food substances. Okay, so they release some enzymes. Okay, they release some enzymes, some digesting enzymes, and they first digest these food substances. Okay, these food substances are broken down outside the body and the broken material is then taken inside the body. It is absorbed through the surface of the body, right? So this is how it happens. The digestion or the breaking down of the material doesn't take place inside the body. As in humans, we eat food, we eat complex food, and that complex food is broken down into simpler materials inside our body only. But in these organisms, like in fungi such as cuscuta, also known as amarvale commonly, These organisms they release enzymes outside their body and digest the food and then absorb it, right? So this is how it happens, and that is why it is said that it depends on the type and availability of food material as well as how it is obtained by the organism. So heterotrophic nutrition actually differs, and it has many types. Like it has parasitic type, saprophytic type, holozoic type. So these are the kind of nutrition that again, these are the kind of nutritions that again take place in heterotrophs. Next up, we have the miracle that is called photosynthesis. So you all might know about photosynthesis, you all might have some idea. So what is photosynthesis? Photosynthesis is a process by which autotrophs take in substances from the outside and convert them into stored forms of energy, right? So autotrophs make their food by a process, and the process is called photosynthesis. So photosynthesis is basically a process by which autotrophs make their food. Now here you can see that a plant is shown, a flower plant is shown, and here how, like, what all substances are actually used in the process called photosynthesis. You can see here sunlight, chlorophyll, carbon dioxide and water. These all things are needed, and then oxygen is released out. Okay, so we'll be studying further in this context. We'll be studying more about photosynthesis in the coming slides. Here you can see that how carbon dioxide is released by humans, and then it is taken up by plants, and the plants again produce food, and then these food are taken in by the humans, and this is a cycle between the plants and the humans. So as you can see, this is a very beautifully they have shown the cycle. Okay, in the presence of light and chlorophyll, carbon dioxide and water are converted to glucose and oxygen. Glucose is then converted to other complex forms called starch, which acts as an energy reserve. This process occurs in special green organelles called chloroplast, which are present inside cells. Chloroplast contain chlorophyll. So as you can see, this is light, this is chlorophyll. It's a very important thing that you need to remember. Chlorophyll is the plant pigment. Okay, so chlorophyll is the plant pigment. It is green in color. Okay, it is green in color. And where is it present? It is present in chloroplasts. Okay, and these light and chlorophyll and carbon dioxide and water, these actually act together, and then it is converted into glucose and oxygen. So as you can see here, this is a very important equation that you need to remember always. Whenever you are talking about photosynthesis, you need to explain this process, this whole process, that carbon dioxide and water in the presence of chlorophyll and sunlight combine together, they react together, and then they form glucose. Okay, and oxygen is given out. So this is a process, this is an equation that you will always need to remember, and you will need to mention whenever you are mentioning photosynthesis. And then this glucose which is produced is again further converted into starch. So starch is basically the stored form of energy. Okay, the stored form of energy. So you will always get a question that in which form is glucose or is energy stored in plants. So you'll write that it is stored in the form of starch. Starch is basically the energy reserve that we have in plants. And what are the steps of photosynthesis? So we'll be going through the steps of the photosynthesis one by one. Step 1, chlorophyll absorbs light energy. So there's this chlorophyll, this is plant pigment, green pigment, which is called chlorophyll, and it absorbs light energy. Then step 2, light energy is converted to chemical energy, and this energy is used to split water molecules into hydrogen and oxygen. So water molecules are again further broken down into hydrogen and oxygen, right? This is how it happens. Then step 3 is carbon dioxide is reduced to carbohydrates. So carbohydrates, what is a carbohydrate? Which form of carbohydrate? Glucose. Okay. So oxygen is also given out, glucose is also given out. For example, desert plants take up carbon dioxide at night and prepare an intermediate which is acted upon by the energy absorbed by the chlorophyll during the day. So you can see in desert plant what happens is they take in carbon dioxide at night. Okay, at night time, during night time desert plants take in carbon dioxide. Okay, they take in carbon dioxide and they have an intermediate. Okay, they form an intermediate. So this intermediate, then again it is acted upon by energy absorbed by the chlorophyll during the day. So during the day what happens? During the day this chlorophyll actually is acted upon. This intermediate is acted upon by energy that is absorbed by chlorophyll and then glucose is produced. So this is how a process that happens in desert plants is discussed here. Okay. Then we have a cross section of leaves that we will be just going through and examining how a leaf cross section looks like and how we can spot the chloroplasts in which the chlorophyll are present, how we can spot them in a leaf. So we can see here the diagram, a leaf diagram is given and there is this transverse section. Okay, a leaf is cut. Okay, in transverse section it is cut. Here the parts of the leaf is also shown. Like you can see here, this is the midrib, this is the lamina or leaf blade, and this is the veins. These are the veins. Okay, these are the veins that are shown here. Now, if you cut this cross section, okay, you cut this cross section and you examine it under a microscope, you will see this structure in the microscope, okay, in which you will see that the outermost layer is a waxy cuticle, okay. This is a protective layer that is present on the leaf. Then below it we have upper epidermis. Then we have chloroplast. Okay, we have this inside it we have chloroplast. These are the cell organelles in which chlorophyll is present. Okay, so chloroplast, these are green in color. So green in color. Okay, these are green in color. We can always say that these are green in color. Then we have the air spaces also. We have certain air spaces in leaves, and these are the guard cells. Okay, these are present in the lowermost surface. Okay, the lower surface of the leaf. We have these guard cells in which we have stomata. Okay, in the guard cells we have stomata. So we'll be talking about stomata. We'll be learning about it, studying about stomata later on, and we'll see how stomata is present in the leaves. Okay, and this is the lower epidermis. So this is the upper epidermis on the upper surface of the leaf, and this is the lower epidermis on the lower surface of the leaf. So you are cutting the leaf in this way. Just imagine this is a leaf and we are just cutting it this way with a blade, and then we are examining this cross section under a microscope. Okay. And what is stomata? As we discussed before, that stomata was present in the lower epidermis. So we'll see what it is. Plants obtain carbon dioxide through small opening on the surface of leaves called stomata. They are present on the surface of leaves, root or stem surfaces. So these you have to when you are asked about stomata, you have to mention this, that carbon dioxide which is required by plants for photosynthesis, that is obtained through small openings on the surface of the leaves called stomata. So these are the small openings on the surface of the leaves. And where are these present? These are present on surface of leaves, root and stem surfaces. Right? So you can see here that how a stomata looks like. The opening of the stomata, you can see these are the guard cells. Okay, these are the bean shaped guard cells, and then outer side you can see the epidermal cells. Okay, the supporting epidermal cells are present on the outer side of the guard cells, and these are the guard cells. So these guard cells open and close. Okay, so they open, and when they open, the carbon dioxide is taken inside, and when they close, there is no scope for carbon dioxide to go inside. And there is also exchange of gases also takes place through the stomata. So whatever waste products that happen, like that, are produced in the plant, the gaseous waste products are also exchanged through the stomata itself, right? So this is also a very crucial apparatus. Is also a very crucial apparatus for excretory mechanism in plants. So stomata is a very important organelle, and you need to learn about it. You need to understand that there is this opening which opens and closes, and through these the exchange of gases take place. Okay, we'll always remember that stomata is for what? Stomata is for exchange of gases. Okay, so exchange of gases takes place in stomata. You can see here the stomatal pore. We have the guard cells with the guard cell nucleus. We have vacuoles also in the guard cells. Okay, and then we have the chloroplast here. Okay, so these all things are present in the stomata, and this is a very, very important apparatus. How do plants obtain carbon dioxide? Stomata are minute openings found on the epidermis of leaves, stems, and other plant organs, as we just discussed. And the carbon dioxide, water vapor, and oxygen are diffused. These all gases diffuse into and out of the internal tissues of the plant through stomata. So stomata is basically a apparatus through which exchange of gases takes place, as we said. And which gases takes place? Carbon dioxide, water vapor, and oxygen. So you need to remember the names of these gases: carbon dioxide, water vapor, and oxygen. These takes place. The transport of these gases takes place. The exchange of gases takes place between the internal tissues of the plant and the environment through stomata. Okay, so these are the openings when photosynthesis is required. They are the openings when photosynthesis is required, leading to loss of water vapor. They remain closed when photosynthesis is not required. Okay, so the stomata remain closed when photosynthesis is not required, and the opening and closing of the pore is the function of the guard cells. As we saw, that guard cells are bean-shaped cells. Okay, they are bean-shaped cells. You can see, beans, the pulses that we eat, right? So bean is one of the pulses, bean shaped. Okay, so these are the bean shaped guard cells that we have. Okay, and the opening and closing is a function of these guard cells. Okay, the opening, and this is the stomata. Okay, this is the stomata. So opening and closing of this pore, the stomatal pore, is basically the function of these guard cells. Okay, the guard cells swell when water flows into them. Okay, so when they absorb water, they swell up, and then they open. Okay, and similarly the pore closes if the guard cells shrink. So when the water is again given out, okay, then they shrink, and when water enters, they open, right? And when the water goes out of the guard cells, they close. So this is a very simultaneous process that is going on in the plants, in the leaves of the plants, and this is how carbon dioxide and the exchange of gases takes place in the plants. Next up, we have an activity. The activity actually is all about, is chlorophyll essential for photosynthesis? So we will perform an activity to prove that whether chlorophyll is essential for photosynthesis or not. For that, we'll be taking a potted plant with variegated leaves. So variegated leaves means that the leaves they'll have patches of different colors. Okay, they'll have green patches and they'll have these yellow patches. So this is known as a variegated leaf. You can very readily find variegated leaves outside. In your garden itself also you can find variegated leaves. So what are these leaves? What will we do with these leaves? We'll take the potted plant with variegated leaves and then keep the plant in a dark room for 3 days. Okay, so we'll keep it in the dark room for 3 days, and then keep the plant in sunlight for about 6 hours. So we'll first keep it for 3 days in dark, and then we'll keep it for 6 hours in sunlight. Then we'll pluck a leaf, okay, leaf of the plant, and mark the green areas. We'll mark the green areas in it and trace them on a sheet of paper. So we'll mark the green areas and we'll trace them on a sheet of paper. Then we'll dip the leaf in boiling water for a few minutes. Okay, we'll dip the leaf in boiling water for a few minutes, and after that we'll immerse it in a beaker containing alcohol. Okay, then carefully we'll place the above beaker in a water bath and heat till the alcohol begins to boil. Okay, and then we'll dip the leaf in a dilute solution of iodine for a few minutes. And then we'll take out the leaf and rinse off the iodine solution. What will we observe? We'll observe that only those regions of the leaf which were green in color turn blue-black. So only these regions perform photosynthesis. Now here we can see that only those regions are blue-black, okay, which were green in color. See? Blue-black, okay, they turn blue-black in the iodine solution, and this is green in color. Okay, so basically what we can understand from this activity is that the photosynthesis only took place when the plant was kept in sunlight for 6 hours. The photosynthesis only took place in the green parts of the plant, and this green color, as we already discussed, that this green color is why is this portion green? Because here we have a pigment called chlorophyll. So we can always see that since the photosynthesis took place only in the greener parts of the leaves, so we can say that chlorophyll is a very necessary thing for photosynthesis. If there is no chlorophyll, like this yellow parts, they do not have chlorophyll, so the photosynthesis did not take place in chlorophyll. So the chlorophyll is a very important pigment, is a very necessary component for photosynthesis, and that can be proven by doing this activity. Next up, we have a question that is carbon dioxide necessary for photosynthesis? So how can we prove that carbon dioxide is a very necessary component for photosynthesis? For doing that, we'll do an exercise, we'll do an activity where we'll take this whole apparatus where a plant is put inside a bell jar, okay, and then so in this we'll take 2 healthy potted plants and then keep them in a dark room for 3 days. Okay, we'll keep them in a dark room for 3 days and then place a watch glass containing potassium hydroxide by the side of one of the plants. Now cover both the plants with separate bell jars. So we can see here, this is how we are going to do it. We'll cover them with separate bell jars and then we'll use Vaseline to seal the bottom of the jars to the glass plates so that the setup is airtight. So we have to make this setup airtight. Now what we will do is we'll keep the plants in sunlight for 2 hours. Okay, we'll keep the plants in sunlight for 2 hours and then we'll pluck a leaf from each plant and check for the presence of starch. Meanwhile, what we'll do is while keeping one of the plants, we'll put a watch glass containing potassium hydroxide with that plant. Okay, and in the other we won't put anything. So what we can do, what we'll observe is the plant which had KOH, like the potassium hydroxide, the formula of potassium hydroxide is KOH. Okay, so the plant which had KOH together with it in the bell jar, the leaves of that plant won't turn blue-black. Okay, the leaves of the plant which had no KOH in the bell jar, they show blue-black color. So with this, what we can conclude is that what we can understand is that KOH, this potassium hydroxide, absorbs all the carbon dioxide. It is a good carbon dioxide absorber, right? So what it does is it absorbs all the carbon dioxide here inside the bell jar. So what happens is these leaves for photosynthesis we need two very important components, right? Carbon dioxide plus water in the presence of sunlight, okay, and chlorophyll. So here in the KOH jar we do not have carbon dioxide. Carbon dioxide is missing. So will photosynthesis take place? Of course, it won't take place in this jar because carbon dioxide is missing. Because the KOH has taken all the carbon dioxide. It has absorbed all the carbon dioxide. But in this one, carbon dioxide is present, right? Carbon dioxide will be present, and that is why the leaves of this plant will turn blue-black in iodine solution, confirming that they produced starch through photosynthesis. So this is how we are going to do it. Then what we can conclude is that carbon dioxide is very, very necessary for photosynthesis. In the previous activity, we were proving that chlorophyll is very necessary. And in this activity, we are again proving that carbon dioxide is very necessary. So here we can see that these four components actually are very, very necessary for doing photosynthesis. The absence of any one of them will actually not perform photosynthesis. Like if any one of these components are absent, okay, then photosynthesis will not take place in the plant. Now again we are talking about sunlight, okay, so we are taking one by one all the components and we are proving that all the four components are very, very important for photosynthesis. Now here we have the component of sunlight. What we will do is the experiment that we will take a potted plant and keep it in a dark place for 2 to 3 days and then cover a part of one of its leaf with the strip of black paper. So here you can see how a strip of black paper has been used to cover a part of the leaf, okay, and then we'll place this leaf in the beaker containing water and boil it for about 10 minutes. We'll take out the leaf and now boil it in alcohol using the water bath for 10 minutes. This removes the chlorophyll. Take out the leaf and wash it under running water. Okay, we'll wash the leaf under running water and then we'll place this leaf in a petri dish and put a few drops of iodine solution onto it. So what we will see is that the area of the leaf which was covered and which did not get sunlight, okay, the area of the leaf which was covered and it did not get sunlight, so this area will not turn blue-black. So here the photosynthesis did not take place. So since the photosynthesis did not take place and there is no starch present, that is why this area will not turn blue-black. But the other area, since it was kept in sunlight for 3 to 4 hours, the other areas had photosynthesis and they will turn blue-black. So what we are seeing here that iodine is an indicator. Okay, iodine is an indicator for starch. Okay, since photosynthesis produces glucose and glucose actually changes into starch, which is the reserve form of energy in plants, so we can always try and figure out whether photosynthesis has taken place in any part of the plant by just doing this iodine test. Okay, so we are performing the iodine test just to find out whether we have starch in that part of the plant or not, and whether that part of the plant had photosynthesis or not. So here, since sunlight did not fall on this part of the plant, there was no photosynthesis and in turn there was no starch present, and that is why we can conclude that sunlight is necessary for photosynthesis, and if any area of the plant is not getting sunlight, then it won't produce starch and hence it won't have photosynthesis in it. Here we will be discussing about the different types of digestive systems for different organisms. So here we are discussing about the single-celled organisms like amoeba. Okay, so how do they digest their food? So they digest their food by taking in food through their entire surface. So they do surface digestion. Like you can see, here's a food particle, and then this amoeba is going and then producing its pseudopodia. Okay, what is pseudopodia? Pseudo means false, and podia. Okay, podia means feet. So it has these false feet in itself, which it produces this false feet and then captures the food molecule and then digests it. It takes the food molecule inside its body through its surface. So that is how they digest their food. But in multicellular high level organisms, that there is a presence of digestive system. So there is a definite system in the higher level organisms to digest food. So we will talking about these nutritional processes in high level organisms like humans in the next video. Thank you so much for staying tuned, and I hope you like this video. Thank you.